Hi, everyone. Welcome to VanBug's first talk of the 2021 season. Today, we are sponsored by the graduate program in bioinformatics at UBC and Simon Fraser University. And we are also sponsored by Genome BC, which is now celebrating 20 years since its founding, Ecoscope at UBC, and also bioinformatics.ca, and Abcelera, and Langara College. Also, we're affiliated with ISCB, as well as other bug groups, bioinformatic user groups throughout Canada, including Montreal and Toronto. And one thing I'd like to mention, we are currently looking for speaker nominations for our fall 2021 to spring 2022 season. If you go to vanbug.org and click suggest a speaker, you can nominate a speaker there, and we'll consider those in our process for getting speakers for the next year's season. Today, went through announcements, and now it's time for us to go to our featured talk. Today, we are pleased to host Dr. Carolina Tropini, who's assistant professor at the Department of Microbiology and Immunology in the School of Biomedical Engineering at UBC. Dr. Tropini conducted her PhD in biophysics at Stanford University under Dr. Casey Wong, where she combined computational and experimental techniques aimed at both investigating bacterial mechanics and morphogenesis. And she went on to study the impact of physical perturbations on microbial communities in the gut during her postdoc at Stanford under Justin Sonnenberg. And at UBC, the Tropini lab currently focuses on how disrupting physical environments can affect microbiota and their hosts at multiple different scales, which involves employing techniques from multiple domains that work to create highly parallel assays aimed at understanding how microbial communities function. Today, your talk is entitled How the Environment Shapes the Microbiota in Health and Disease. And with that, I will pass it off to Dr. Tropini.
Thank you, Will, and thanks so much for having me here. It's I'm really excited to be chatting with you all, and even though we're in 2021, we're still doing our Zoom talks, and I would really like to encourage people to interrupt me when you have questions, and to really stop me throughout the talk. I really hope that this doesn't have to be a monologue, and I really appreciate feedback throughout the talk. So it'd be great to have this as a conversation.
Yes. And before you start, we will be monitoring questions through our chat. And also, if you want to speak out your question, please use the raise hand feature, and one of me or one of our moderators will interrupt Carolina.
Great. That sounds probably more orderly. As Will mentioned, my background is in physics, and I came to the field of the microbiota really trying to understand how factors in the physical environment, so things like pH, temperature, osmolality, physical forces affect the way that these microbes can organize together. And one of the things that was really obvious to me early on, starting in this field, was the fact that the type of questions that are ecological in nature that get thought about for microorganisms somehow are very different from macroorganisms. So if I had to ask you what determines the ability of an organism to colonize a habitat, and if I asked you this for a plant as opposed to for an E. coli cell, you would likely tell me different things. For microorganisms, we tend to think about how biochemical interactions will affect the way that things can, say, colonize a host. But if I had to tell you about a plant, you'd likely be telling me about the environment. So that if I told you that I would like to transplant a cactus into the tundra, you would right away have an understanding and a hypothesis as to what might happen. We have this understanding that different plants will require different levels of water and different temperatures for their ability to survive. But somehow this type of understanding doesn't actually translate very directly to microorganisms, even though they really have the same requirements than any organism does to be in a habitat that supports its growth. Why this is important when we think about microbial therapies, such as fecal microbiota transplants or even probiotics, is the fact that when we're trying to have a new organism embed within ourselves, we need to make sure that we can actually sustain its growth and its life. Why this is also very relevant to our thinking about real life organisms and how they're being colonized is the fact that even in a healthy state, our gut environment is composed of very different sub-environments. Here is just a bit of a cartoon depiction of what happens throughout the gut. Here you can see at the top we have a mouse gut, but this is true for any animal gut. There's different environments. And you can imagine that these environments are set up primarily by physiological processes that happen in the host, in the animal host. For example, we have the stomach, which sets up an acidic environment. And this acidic environment over the length of the digestive tract becomes affected. In fact, it tends to become more basic and neutral up until towards the end of the colon. Also, bacterial fermentation reduces this pH. And the host balances this out by producing bicarbonate. And it does an exchange of acidic compounds that are produced by the microbiota in order to offset this balance. There's also very large changes in bacterial load. And importantly, this is something that is very key for the host, because as hosts, we want to make sure that we can get the easy nutrients. So the sugar, the proteins, the amino acids, we want to be able to absorb them before the bacteria start degrading them. So up in the more small intestine, we're having a large production of antimicrobial peptides that keep the microbiota at bay. But then in the end part of the colon, we actually start taking advantage of the fermentation of the microbiota to use other compounds that are useful for our not only metabolism, but also for our health. There's also gradients in oxygen. Oxygen tension is very high in the small intestine, but there's no oxygen in the distal colon. Now, all of these different environments that arise from these differences in physical and biochemical parameters mean that the microbiota that is present at certain points in the gut is going to be very different from the one in other parts of the gut. So for example, earlier on in the small intestine, we have bacteria that are very good at dealing with oxygenated environments and that are good at dealing with our immune system. While further downstream, we have bacteria that are very good at degrading our fiber. And this type of landscape and this type of biogeography is not only present at this large scale at the organ level, it's really present also at the scale of the bacteria themselves, so this micron scale. What I'm showing you here is a section of a gut. This one is in particular, it's a mouse gut, but this would be very similar to what we would see in the case of a biopsy. So what you can appreciate at the bottom, these are the host epithelial cells, the colonocytes. You also have goblet cells that are secreting this mucus that here we have false labeled in green. And we have this very interesting interaction between our host cells and our microbiota. Because on one side, we want to keep it at bay. We want to keep it at a safe distance because these microbes, given the opportunity, they will start consuming our cells and they will try to invade in the case of pathogenic bacteria. So the mucus provides this physical mechanical barrier that prevents the microbes from penetrating. But we also want to keep the microbiota there. So if given a choice, epithelial cells will actually prefer consuming short-chain fatty acids. These are some of the products that are produced by the microbiota, rather than eating glucose. We're so well adapted to this microbiota that the interaction between these cells and our own cells is really, really tied together. And we have evolved with these bacteria and viruses and fungi ever since animals had a gut, where we're talking about hundreds of millions of years of this continued interaction. And one thing that, if you're going to remember a couple of things that throughout the talk, this is, I feel like from a practical perspective, is potentially the most important one. If you don't eat your fiber, these are the large red structures that stain really well because they have a lot of DNA in them. This is a DAPI stain. The mucus layer becomes much thinner because now the microbes don't have things to actually eat. And they will start eating the mucus because the mucus is made on purpose to have these glycans that the microbes can actually degrade. And what happens if you degrade your mucus is that now you have less of a protective barrier. And this can allow pathogens to penetrate much more easily. So this is a little bit of the picture into how different levels of the gut are really tightly integrated with our microbiota and with the environment that this part of the gut affords the microbiota. And as you can see, if you merge both pictures together, you really get to see a sense that this gut environment presents very different environments. And as a bacterium, if you were to go through the digestive tract, you would really want to find your specific niche where you can grow optimally and where you're actually able to give rise to a progeny and where you're going to be most likely to not be thrown out. You know, we very often analyze the stool into thinking about how the microbiota functions. But from the perspective of the bacteria that actually make up our microbiota, those are the ones that are being thrown out. So we care very much about how bacteria behave and function within the context of their biologically important function. This now, I've told you that the gut has all these physical environments that are already set up in health. And as you might imagine, what happens during disease is that these physical environments change. And from the perspective of the microbiota, this is changing the rules of the game. The microbiota occupies this very saturated community that is very, very competitive. There's very little nutrients and the competition is there at all times. So what happens, for example, when there's change peristalsis or flow is that the types of microbes that are going to be able to survive will be different. Even changes in environment, like changes in temperature that might happen during a fever or by the consumption of anti-inflammatory drugs will have a really big impact on the microbes that are able to survive within this really competitive environment. We also see changes in pH that are caused by inflammatory bowel disease. They're sometimes caused by cancer. So you can have these local pockets that have a different pH environment. And that are also, of course, caused by antacids. So we very much, these are a very prevalent type of drugs that people routinely take. And this will change the natural equilibrium of the physical pH that happens in the gut. Another measure of physical environment that we routinely do to try to assess how this world in which our bacteria are living in is changing is osmolality. So this is really, in some sense, if you want to think about it from the macro scale world, it's the measure of, for example, salinity of a lake. And this will very much change the type of organisms that are able to live in this environment. We exploit the use of osmolality by, in the case of laxatives. And also this is what happens for people that have a lactose intolerant or have celiac disease. Whatever compounds are not being absorbed, they will increase the local concentration of molecules and draw water out from the epithelium, therefore increasing the flow that happens through the gut. All of these different physical parameters are nothing new. We've known about them for so long that in our own experiments, we fix them all. Whenever we grow bacteria, we grow them at the optimal body temperature at 37 degrees. We have made our media in a way that they actually match the osmolality of the gut by adding different types of salts, even though we didn't even know that we were optimizing for osmolality, but that's what we've led to. We changed the oxidant tension so that bacteria that are good at growing in oxygenated conditions are able to grow as quickly as they can. What I would like to drive home here, though, is the fact that these different changes in the physical environment can have really long-lasting effects. At the macro scale, this is now becoming painfully obvious with global warming. In global warming, if you think about it, it's a really small change in temperature that we're dealing with. We're dealing with a couple of degrees of temperature change, but this is leading to 1 million species to go extinct. If you now think about what this means at the micro scale, is something akin to a fever. And a fever is also in the order of the same change in temperature. But while our macro scale animals have generation times that are in the order of years, our micro scale organisms have life cycles that are in the order of tens of minutes. So if you have a fever for a couple of days, this is able to wipe out a very large fraction of your population if it's not able to either adapt to this changed temperature or if it's not able to survive the changed competition. And I see that there are some questions in the chat. Maybe I'll wait for Will to let me know.
Yeah. So I believe one of the questions has already been answered in your previous slide, but I think one thing that's still a question from Raul is fiber one of the critical components of the gut for cells and microbes? I think that wasn't.
Thanks, Raul, for that question. There's a lot of emphasis being put on healthy food and fiber. The reason why fiber is so important for us is the fact that we have evolved eating a lot of it. The standard dose that people say that people should take is about 30 grams per day. And that's ludicrous compared to what our ancestors ate. Our ancestors ate upwards of 100, 150 grams of fiber. There's nothing that is particularly special about fiber, except the fact that because we evolved eating fiber, we also evolved expecting the products that are made from fiber fermentation that our microbes produce. So then what happens is that very quickly, in the span of less than a century, we change our diet to having diet that is very low in fiber, foods that are very highly processed, much easier for us to consume, a lot more sugar. And these are all compounds that are fine on their own. It's just that they're not good for our health because we're not adapted to them. Unlike us that have generation times of 20 to 30 years, our microbes have these short generation times, which means that over a few weeks, they're able to change. They're able to adapt to these different diets, and they will start producing things that this new diets allow us to actually make, which is fine for them, but it's not fine for us. Basically, we need some of the compounds that are made by fiber fermentation to reduce inflammation. We need them for correct brain development. We need them for having the right balance between us and our microbiota and the immune system. So it's really where I see it is in the eye of evolution, of how we have evolved eating fiber, and we have not caught up with the changes in our diet that we've taken. So fiber itself is one of these things that we have evolved with. This was a kind of a very long-winded answer, but I thought it was important to put it into context.
Yes. The other question, I think I could reshape it here. So, are things like pH, oxygen, and other conditions more regulated by the organism themselves, or is it more a function of the microbiome at that state, or which one is really? How do you think about this?
Yeah, that's a great question. The answer, like with anything in biology, is that the most complicated way it could happen is likely the way that it does happen. The host definitely plays a very important role in setting up this equilibrium, but the cells themselves will be able to reinforce their environment. And unfortunately, the cells that are best at surviving in disrupted environments are the pathogens. That's how the pathogens are not good at degrading fibers and doing some of the complex metabolism that we would like to keep in our relationship with our microbes. But a lot of pathogens such as C. diff and Vibrio cholera and pathogenic E. coli will trigger diarrhea. They will trigger a physical dysfunction that then the microbiota that we host is not able to sustain. And so basically, instead of trying to compete on the metabolic level where they're not able to surpass the microbiota that is our commensal microbiota, they will change the environment so that they are at an evolutionary advantage, and even less than evolutionary, there's a local advantage to be able to take hold and grow quickly. So I guess the general answer is that it happens in both ways. And when the host is not able to control this environment tightly, then that's when disease starts occurring.
Thank you.
So in thinking about these different perturbations, and we kind of talked about the importance of these time scales, and as well as this length scales that happen, right? You can imagine with a fever that it's not just what happens at the host level that matters because the host is having these changes in temperature, there's also something that matters a lot at the way that even molecules are going to be interacting together because the temperature is changed. So when we think about the relevant scales of physical perturbation, they really go across all of the different scales, from subcellular processes, even molecular scales, all the way to the scale of the host, as well as the scales in between. And from the perspective of thinking about how to tackle these questions scientifically, that means that we cannot have individual measurements. We really need to have measurements that are multi-scale, both in the dimensionality part, but also in the time scale domain. And another thing that this means is that sequencing alone will not be able to tell us the answer, because sequencing, for the most part, will target our shorter length scales that will tell us about bacterial communities, and it'll tell us about how expression levels might be changing. But we need to also have information about how the interface, how this mucus layer might be affected by, say, having a fever or having another type of malfunction in our organs. And we also need to understand how the host behaves. So there's a lot of different layers that need to be unpacked together and then rebuilt up. And this, from the computational perspective, provides a very exciting challenge, because these measurements are often very disparate, and our goal is to really bring them together to have this deeper understanding. Today, what we're going to talk about is this idea of how osmolality changes the way that our microbes are able to survive, and how this in turn affect our own health. You probably have heard of osmolarity, and osmolarity is a measure of the number of moles per mass or per volume. In the case of osmolality, this is a even more fundamental measure. This is the number of molecules that are present in a volume. And the reason why at a physical level we care about osmolality is because of the physical pressure that different molecules that are moving around because of thermal motion that is exerting on a permeable membrane. And importantly, this thermal motion is very important for bacteria, because bacteria are very highly pressurized. In the case of E. coli, the pressure is about an atmosphere. This is in the order of what you might imagine a normal road bike to have. But in the case of other bacteria, it can be up to hundreds of pascals, hundreds of atmospheres of pressure. So why this matters from the perspective of osmolality is that if you have a new molecule that comes across and that is not able to permeate through the membrane and actually equilibrate this osmolality, it means that the only way that the cells or this entities are able to equilibrate is by having water diffuse out of the cells. This happens to our own cells as it happens to our bacterial cells, but the bacterial cells are more highly affected because of this larger turgor pressure. And as water flows out, the cells shrink. And this is something that happens very quickly, in the order of milliseconds, this equilibration. But this causes the cells to halt a lot of different physiological responses. And the cell at this point needs to be able to reinflate to work properly. So there's going to be a cascade of different transcriptional responses that will happen at the level of how the cells are going to be working. And one of the things that you might wonder is how often these type of perturbations might happen. You know, if you're thinking about when this happens in nature at a large scale, you might imagine that at estuaries where different rivers are meeting with the sea, cells may be shocked osmotically. But it turns out that this happens in our gut very regularly. Not only because, as I explained, if you're eating something that you're not able to absorb, and there's an intolerance of sorts, or there's something like inflammatory bowel disease, and so there's a lot of these unabsorbed molecules. But this happens also in the case of laxatives. And laxatives are very, very highly used in the North American population. So Miralax, which in Canada is called Restoralax, is the top 2 digestive remedy in the United States. Here you see that about 5 million kilograms per year are sold. This number is humongous if you think that the standard dose is 17 grams. So in terms of our collective microbiota, this type of compounds and this type of osmotic perturbations are things that our microbiota experiences very regularly. And what we wanted to understand was how acute this type of osmotic perturbations really are. When we think about taking a laxative or having malabsorption, we generally think about it being done by the time that we start feeling better. But we wanted to really understand from the microbiota perspective whether this was the case. And to do so, we started taking an approach that allowed us to really separate some of the components that will be affecting this question. As I've shown you before, there's different levels at which these physical perturbations will affect our health, and we wanted to be able to tease those apart. So we looked at this non-abiotic models of disease. So these are mouse models that are derived to be germ-free. So we're in the womb, our understanding is that there are no microbes, and we're able to take these mice and have them not have any microbes in them at all. So before the act of birth, when we're first seeded with our microbiota, we can collect their wombs and produce these colonies that do not have any microbes in them. And then what's really powerful at this point is that then we can start colonizing this germ-free mice with individual microbes. And we can then really get at the mechanism for how different things will be happening in our microbiota. And at the same time, we can also take complex microbiotas that may come from patients, and we can start to recapitulate some of the effects that are due to the microbiota with these types of mice. So from the perspective of this osmotic perturbation, we wanted to have a mild perturbation that was more similar to a chronic exposure into these laxatives rather than something more dramatic, such as a colonoscopy prep. So these mice were exposed to Miralax, which I'm going to refer to now as PEG. This is polyethylene glycol in the water. And this was not forced on them, so they could drink as little or as much as they wanted to. And they were exposed to this for a period of 6 days and then allowed to recover with, again, with normal water. One of the things here that we didn't know at the start was what to think that would happen in terms of the gut osmolality. What you could think is that because there's this non-absorbed solute that is happening throughout the gut, we know that this causes increased motility because of water flowing out of the epithelium. But this itself could be balancing the osmolality, and we weren't sure whether we could be going in either direction of normal osmolality. So we actually measured this in the intestinal contents. And what we found is that during the time of PEG treatment, the osmolality was indeed increased. So here you can see that a normal gut osmolality is in the order of about 450 milliosmoles per kilogram, and it almost doubles during the time of PEG treatment. And one thing that biologically I think is really beautiful is the fact that if you look at the blood osmolality, how tightly it's kept, this really indicates how important this type of factors are for us. If blood osmolality changes even by a couple of percents, this would mean death for us. So from the perspective of the host, even though these mice are over time, because they have this Miralax in the water, are becoming dehydrated, their blood osmolality is still kept really tightly in check. And interestingly, this osmolality level, which is about 350 milliosmol per kilogram, is perfectly matched to that of PBS. Once again, one of these things that we didn't necessarily directly optimize for, but that within our scientific community, we optimized so that it would be perfect for growing our cells. So as you can see here, there's this change in osmolality, and I see Will again, so I imagine there might be a question.
Yeah, so more questions from Brahim and Raul. I'll just start with Brahim first. So is this method for like using osmotic perturbation for taking out, basically knocking out a microbiome in a mouse, does that make sense to do in other organisms? Could it be done in humans?
Well, so what I'm going to show you actually, that was a perfect segue. It doesn't knock out a microbiota. What it changes is who's there. So here we're looking at a colony forming units, which means that the culturable microbes. So here it's basically like in 1 mil of intestinal contents, we get about 10 to the 10 microbes that are anaerobic, and we get about 10 to the 8 microbes that are aerobic. This is probably a slight, or it's a bit of an underestimate because we can't culture everything, but it kind of gives you a sense of the fact that if anything during the PEG treatment, so this is highlighted in yellow, we get a mild increase in microbes. And so this is important to know that if this was a colonoscopy prep, we actually would be flushing things out. But during the time that the motility is increased, but it's not a tsunami, you're just changing who's able to survive in this environment. You're not significantly changing how much food there is there for them to eat. So you're basically, as you're changing the rules of the game, it just means that certain players are going to be able to play better than others are. So when you think about cleaning out your microbiota, laxative is really not the way to do it, unless you're taking such high amounts that you're causing the more tsunami part of the picture.
Yeah. Well, I think there's one more general question here. So are we born with bacteria inside our gut? This is from Lorenzo Jose. And also, what role does aging play in changing that, if at all?
Yeah, great question. Thanks for that, Lorenzo. We're not born with a microbiota in the womb, but when the first thing that we see through our birth canal are bacteria. And one of the things that is really, again, like this beautiful interaction between us and our microbes is the fact that the maternal birth canal prepares for birth by changing the microbiota in a way that we're mostly at the beginning coated by bacteria that are very good at breaking down our maternal milk products. So we're so tightly connected with these microbes that when babies are breastfeeding, about 1/4 of the dry weight of maternal milk is compounds that the baby cannot consume. They're just these very complex sugars that are there for the microbiota to eat. Even I think more surprising, if you look at mothers that are malnourished, these components are still there in the milk. They have no fat. It's basically their milk is liquid, but it still has these sugars, which really underscores the importance of keeping our microbiota healthy and built up correctly. If you notice, kids put everything in their mouth, and we're really starting to believe that it's the way in which we start seeding ourselves with microbes around us. Of course, the maternal birth canal microbiota and the fecal microbiota during the birth process, of course, gets very messy and everything gets mixed in there. But it's very, very important in our first seeding. And for babies that are born via C-section, sometimes they have a microbiota that is more similar to the skin microbiota of the surgeon that performed the surgery than their own maternal microbiota. And there's a lot now that we're starting to understand about the importance of being seeded with the correct microbiota, which really means with a microbiota that our body is expecting starting out. This microbiota changes very strongly with age. So you can imagine that when you're just fed with breast milk, this microbiota is not very diverse. But it's a microbiota that protects you against certain types of pathogens. So for babies that are born prematurely, a big problem that can arise is the fact that there are some pathogens that can get in. But if these babies have their more normal microbiota, they can fend them off. And this seems to be really important. There's some new therapies that are coming from the perspective of NICUs to make sure that that this protective microbiota is there to help against pathogens. And again, like, it's not that the microbiota is particularly generous. The microbiota wants to keep itself safe from pathogens, but having it embedded correctly also protects us because we're these symbionts that are acting together. And over time, what we see with aging is that our microbiota changes in a way that tends to promote bacteria that are better at dealing with oxygen. So it almost seems like the microbiota is becoming more oxygenated, which is something that tends to be correlated with the disease. So with the diseases that disrupt the mucus layer, more oxygen comes from the epithelial layer into our gut. So overall, the microbiota tends to become a little bit less diverse over the years, potentially also because the diet worsens with age.
Thank you. And I think one last question from Raul. So we've covered, so laxatives don't necessarily flush out unless you're taking microbiota, unless you're taking insane amounts.
Right.
Right. And then in the case of a colonoscopy, regenerating this microbiome in your gut, is that like usually a fairly regular process?
We'll talk more about whether chronic versus acute things are better. And I actually, from the perspective of taking a colonoscopy, there's some data that the majority of microbiota recovers because it's such a quick flush out that as long as you have pockets in there, they're able to build back up. You still have a chance for the microbiota to recover pretty normally. It is, in some cases, this is not generally true. There are some species that are going to be at very low abundance that stochastically will be depleted. But that seems to actually be less problematic than doing things chronically. Because as you'll see, and I don't want to give one of my punchlines away, but as you'll see, when you have a changed environment chronically, it's a little bit like the global warming. It's not if you have a 1 year that is really high temperature that you have species go extinct. It's when you have multiple years in a row that this environment is changed. So that the chronic effects, as you'll see also in our data, can have much more long-term effects.
Thank you.
And now we'll tell you about these long-term effects. So one of the things you might imagine about thinking about this physical change environment, as we talked about, you can have these changes that are transient, and we see this in a way that we can predict. For example, we can predict that gamma proteobacteria, these are bacteria that have a lot of genes that are involved in stress responses, your typical E. coli, Proteus, things that are very good at handling perturbations and also living in the environment, are going to be bacteria that bloom during the time of disruption. And one of the things that's interesting at the bioinformatic level, that what we found is that when a family of bacteria was responding positively to the perturbation, it was increasingly likely that a bacterium within the same class would actually be having the opposite behavior. So we see a lot within the phylogenetic tree, we see a lot of these paired behaviors that seem to really indicate that there's niches that open up that then can be exploited by other members that have the ability of filling them up. But importantly, as I alluded to, we also see effects that are long-term. And here I would like to focus on two specific families of bacteria. These are bacteria that are very common. In fact, they come from a human donor. So these are so-called humanized mice that started out germ-free and then got inoculated with a human microbiota. Everyone, all 50 people that are present in this call have Bacteroideaceae, and about probably 5 or 6 people have Moribaculaceae. You can see that these are very strong colonizers of this microbiota, with Moribaculaceae starting out at almost 50% of the microbiota. So really half of the microbiota in this community is Moribaculaceae. What we found is that during the PEG treatment, Moribaculaceae became undetectable. And even more surprisingly, when we took away the perturbation, it just never came back. This was a 6-day course of a laxative at low doses. What we were really surprised to see was that we could actually manipulate 50% of the microbiota and make it disappear. And to the point not only of making it disappear, but really changing the membership of our community. So as you can see here, during the time that Moribaculaceae go away, Bacteroideaceae has a new niche that it can fill into. So once again, this is one of the matching pairs. Bacteroideaceae are actually quite similar to Moribaculaceae, but they're able to get into this niche because they just do better enough. Not only is the fact that we're talking about billions of organisms that are being depleted, just through the course of these laxatives, but also we're really modifying the ecological system that is present in this microbiota, because we're actually taking away an entire family of organisms. And again, if you think about it from the macro scale, it's as if from an ecosystem, you took away all primates or all felines. This is a major change. And it's something that we really wanted to understand what the implications of this were in the way that our microbiota is able to function following this perturbation. One of the things that we wanted to understand was whether this was something specific about the fact that our microbiota, it was a human microbiota in mice, and whether this had something to do with the mismatch of these species together. Because of course, a human microbiota is adapted to their human host, not to their mouse host. So we also repeated these experiments with conventional mice, and these families are also present in a conventional microbiota. And we found the same behavior, that very consistently we were able to deplete Mucin-utilizing, and we didn't recover it back even after months of following the SPEC treatment. Of course, now the question is, how are we leading to this disappearance of an entire family? And one of the really advantageous things of thinking about how the physical environment impacts this highly biological system is the fact that we can separate the effects of the host from the effects of the physical environment. And we can do this by testing the systems in vitro. So if the bacteria are going to be affected by the physical environment, this needs to happen also in our test tubes. Needs to be something that holds true in any type of host or non-host situation. So what we can do is that we can grow our individual isolates with different types of osmolytes and look at how their growth changes. So here we do this measurements by looking at optical density, here named OD, and we'll look at over time what happens. And typical with these bacteria or these microorganisms, we see that the optical density increases and then it reaches saturation. So with different levels, we can then quantify the growth rate looking at the steepest part of the curve, as well as the maximum capacity that these bacteria are able to occur. And what I'm going to be showing you in the next graphs is a measure of the maximal growth rate in changing environments. One thing that also I'd like to note is that we use different types of osmolytes to measure these physical characteristics, because as you might imagine is that if you're in a pool of sugary water, this is going to affect you differently than if you're in a pool of salts. So we want to separate things that the bacteria can actually feed on, as opposed to things that are going to be primarily osmotic in nature. And importantly, there might be some factors that actually can affect the cells differently that go beyond the effect of just changes in osmolality. And here's what happened with our Mucin-utilizing isolates. We see that as we expect, there's this very strong decrease in growth rate with increasing osmolality. Again, we know that we've optimized our media to be as good as it can be. But interestingly, there's no growth happening at levels that are far below the levels that we measure in vivo during the PEG treatment. And importantly, as I mentioned, this is something that's true for all bacteria, but it doesn't matter that you're worse off as long as you can keep up with the competition. So in the case of Mucin-utilizing versus the Bacteroideaceae, and Bacteroides the iota omicron is one of these members of the Bacteroideaceae family, Bacteroideaceae can actually grow as opposed to not being able to grow at all. And so even though it's impaired, it's able to do better in the situation, actually survive. We also see indications that the PEG treatment might have effects that go beyond these changes in osmolality. Specifically, we know that in the lab, we use PEG to precipitate DNA and precipitate proteins. So we think that it also has effects that are bactericidal by modifying potentially the membranes. So here, basically, what I've told you is that also in the absence of a host, Microbiology seems to be very sensitive to this physical environment, to increases in osmolality, more than other bacteria that would be present in these communities. And now 3 members in my lab are currently investigating these results more. So Diana Pepin, Sarah Poppel, and Bakfid and Ginn are trying to understand from different types of organisms that are present in the microbiota, what leads to the resilience or the lack of resilience of these microbes. So the microbiota is not resilient to osmotic perturbation. We see these large disappearance and even extinction events. From the perspective of thinking about just the physical environment, we can think about how increased osmolality impairs the bacterial growth. And we can identify specific stress genes that are going to be overrepresented and also overexpressed during our osmotic perturbation. But importantly, we also need to think about these off-target effects. With things like antibiotics, we often think about how we may be affecting other members of the microbiota, how there's going to be things that are not going to be directly related to the resistance of the microbes to the antibiotics itself. And osmotic perturbations or physical perturbations in general are no different. In fact, they're so broad spectrum, if you will, that they will affect things that are completely unrelated to the osmotic changes itself. So one of the things that happens, for example, with having increased osmolality is the fact that you have increased motility through the digestive tract, to the point of even having diarrhea. And so this causes changes that are completely orthogonal to the osmolality itself. And one of the things that we wanted to investigate from this perspective was indeed the mucus and how the mucosal lining will be affected here. So what we see here, kind of going back again to our initial picture of a healthy microbiota being separated from the host by this thick mucus layer. And we wanted to understand what happened during the time of PEG treatment. Very surprisingly, we found that the PEG treatment completely depleted this mucus layer and detached it from the intestinal lining. I won't have time to show you this, but this leads to changes in the immune system. There's specific molecules that can become attacked by our adaptive immune system that get targeted by both IgA and IgG. We see changes in the way that different immunoglobulins are generally being produced, as well as changes in the innate immune system that we think that are due to these changes in mucus. Importantly, this mucus layer recovers in about a week. So we can quantify the thick part of the mucus layer, and we can see that it recovers to original levels. But we find that the overall levels are not completely represented until about 2 weeks after the perturbation is done. And as the last question indicated, this is potentially going to have effect on bacteria that can degrade the mucus. So a very common bacterium that is mainly a mucus consumer is Akkermansia of the Microbial family. And we did find that Akkermansia during this time of osmotic treatment becomes much reduced in levels, and in fact follows this very similar dynamics as the mucus itself does. So you might also wonder the question of what about bacteria that are able to choose between using mucus compounds for food as well as other compounds. So to answer this question, we looked at a defined community that had bacteria that are able to switch between mucus consumption and consuming other molecules, such as our Bacteroides theta iota micron. And we did find that during PEG treatment, Bacteroides switches off from using mucus as a primary food source, and it starts upregulating other types of glycoside hydrolases that are involved in the breakdown of these complex sugars. So from the perspective of what happens during the PEG treatment, we have this completely orthogonal modification to our environment, and that comes from the form of how the mucus change. And we find that both mucus consuming microbes, as well as this mucus utilization genes are going to be reduced or downregulated. And I wanted to end a little bit on a positive note in thinking about how we can actually restore some of these changes. So fortunately, the mucus comes back, but we wanted to understand whether we can actually restore some of the members of the microbiota that are lost, specifically this Microbiology family. And one of the questions that we asked was, can it be reintroduced in the environment, and will that allow it to recolonize? So with mice, there's a very easy way of doing fecal microbiota transplants because mice are coprophagic, meaning that they eat their own and each other's stools. Again, I apologize. All of this is happening over lunch. We've just been talking about diarrhea and fecal microbiota transplants, but unfortunately, for better or for worse, I've become completely adapted to this type of discourse at any time of day. But what we really wanted to understand was whether by adding this control microbiota that does have Mucin-utilizing, whether we could actually get it back in. And for us, this was a bit of a tricky question, because one of the things that we realized is that during the time of equilibration, the microbiota had actually become, as far as we could tell, saturated. It was, again, back at the original levels. There was bacteriaceae that filled back up this niche. And so we weren't really sure what to expect. We added this control microbiota to the cage, and we were super excited to find out that Mucin-utilizing is indeed able to recolonize. And we're at this time of re-establishment, it's also able to reduce the overall levels of the bacteriaceae. So it's really able to form this type of niche and regain it, which is something that was for us very, very not obvious. And one of the things that we found over time with Mucin-utilizing is that they seem to just be very delicate organisms, in the sense that they're very affected by antibiotics, they're affected by changes in pH, they're obviously affected by this type of osmolytes. And we wanted to have a better understanding as to how important it is for us as humans. You know, all these experiments were done in mice, and given that this was is also a very prevalent human commensal, we wanted to understand more about it. And this gets us back into thinking about microbiota ecology. And one of the things that we know now very well about our microbiota is how we've actually already been doing engineering on it, just because of our changed lifestyle. If you look at the microbiota across different populations, it's very different, and it very much separates on the axis of how industrialized a population is. So when we look at traditional populations across the world, we find that microbiota it's much more highly diverse, and that has species that have been lost in the industrialized microbiota. The industrialized microbiota tends to be much less diverse, and this lower diversity tends to also correlate with this increase in inflammatory diseases and in so-called Western diseases that our ancestors just didn't know about. We wanted to understand where S24/7 laid in this picture. And as I mentioned, about 10% of industrialized humans have it in their microbiota, but it's very, very prevalent in the guts of the many different mammals. In fact, it's one of the most prevalent bacteria that is associated with warm-blooded animals. And what we had access to, which was very exciting, was a data set from the Hadza gut. So the Hadza are a tribal community that is traditional in Tanzania. And we were astounded to find that in the Hadza, it's present between 65 to 90% prevalence, depending on the season. And even more excitingly, some collaborators, this is a study from Maria Dominguez Bello's group, they found that also in the Amerindian, so now we're in a completely different continent, Mucin-utilizing are the main distinguishing group between Yanomami's group that are exposed to urban life versus ones that are still in the tribal population. And one of the things that they're also finding is that even tribal populations that have a medical post have a much lower incidence of S24/7. Work from other groups that have been looking at the effect of other modern diseases such as diabetes on and effects on health have been also showing that the absence of Mucin-utilizing is very tightly linked to worse disease outcomes, at least in these animal models. So we're very interested in understanding whether, given that we are able to reintroduce it, whether there's something that is very special about S24/7 in the way that interacts with our immune system that allows it to be able to repopulate, and as well as how it affects other parts of our health. Of course, it's not enough to know that it's easy for it to disappear. it doesn't necessarily mean that it's going to be important for our health, but we're really starting to accumulate a lot of evidence that it's one of these vanishing taxa, and that it's something that we might actually want to keep in us. So here to wrap things up, what I've told you today is about how the physical environment plays this really important role that some of the time can supersede the effect of biochemical interactions. Because if you're not able to grow in an environment, it doesn't matter who else is around you. This is going to be, in some cases, the limiting step that will allow you to survive or not to survive. I've also told you how with a physical perturbation, it's not enough to do sequencing, and you really need to change the perspective from looking at the multiple timescales and length scales to understand the impact of these perturbations. And from the perspective of Microbiology, it becomes important that we not only think about how easy it is for it to go away, but also think about ways in which we can reintroduce it. So in the case of when we've seen with the re-exposure and it coming back, it's important to know that we have to have this re-exposure for it to happen. And unfortunately, in the way that we have changed our society, we have lost the natural reservoir for a lot of these microbes. This, of course, has meant for us that we've been able to live a lot longer because we're not contaminated by pathogens as much as some of the more primitive populations were. But at the same time, this needs to be a trade-off because these are organisms that we have evolved with and our immune system expects to be interacting with. So I think that as we go towards personalized medicine, we really need to keep both sides of the balance there. We're very excited about Microbiology as a potential probiotic species. So a lot of the work in our lab is devoted to understand how we can understand this organism better, as well as how we can understand its function with our health. And to end, I'd like to thank the people that have been involved with this work. So today I told you specifically about work that 3 people in the lab had done. These are Diana Pepin, Bakfin Yin, and Sarah Popple, that is now in our collaborative reading lab, the Osborne lab at UBC. And it's been a bit of a really exciting and crazy 2 years between the pandemic and everything, and it's been really incredible to work with a really talented group of people that have helped to set up the lab and have really made this the start of our lab a really exciting and wonderful experience. And of course, we're very thankful for our sponsors and our funders for actually giving us the funds to do this research. And thanks everyone for listening and for your questions. It was really nice to chat with you all.
All right, thank you, Dr. Tropini. Do you have time for a few more questions from our audience?
Yeah, I'd be happy to.
Okay, great. So thank you for the talk. It was very, very interesting, and I like the whole arc that you've laid out here. So 2 questions from Phil Richmond. So
I can ask them.
Yeah, you can ask them. Yeah. Go ahead.
First of all, very nice talk. Very, the microbiome is definitely a new frontier for a lot of us, but really exciting work that you guys are doing. I guess my 2 questions, I'll just try and be quick here. The first, which I think you alluded to on that last slide, is that there's this question about whether probiotics can actually reconstitute the gut microbiome. And if you've shown this quite convincingly in mice, I guess the question is, would it also work in humans? And is there something special about the packaging or the delivery of a probiotic, or is it simply just slow modulation through something like a change in diet that you would need to get that reconstitution?
Excellent question, and this is going to be very dependent on the microbe that you're trying to embed in the microbiota. Some of the things that are very important in recolonization is, first of all, that the microbe is alive, and unfortunately for probiotics, this is often not the case. So in the previous lab that we're in, we went through some testing of probiotics and actually found that not only are they not the species that they say they are, but oftentimes they just don't grow. So there's a big market around probiotics. And I think just like you wouldn't just add a new species into Yellowstone, and how if you add wolves to Yellowstone, you're going to have unintended consequences. We need to think about the system in the same way. And in some sense, like when I think about personalized medicine, I think that a probably safer way to deal with this is going to be for people to save their own microbiota. This is a microbiota that our immune system has already talked to and has figured out ways to accept and be in good contact with. So what I imagine that the field will be moving more likely towards, instead of individual species, is going to be to storing some of our microbes. The question, though, is that for the microbes that we're losing, this may not be enough. So I think that some of the changes to really be long-lasting, they might have to happen in childhood, so that we should think about the effects that every generation has in losing these microbes. The Sonnenberg lab had done these beautiful experiments showing that with an industrialized diet, over time, the mice would become less and less diverse. And over each generation, these effects were compounding. So it might be that, let's say with Moribaculaceae, one of the things that we need to do is to try to reintroduce it. And if we're not able to reintroduce it to a host that has never seen it, we might need to do it at a time when the host is permissive. So there's really interesting ecological things that come up. For example, there can be intra-strain competition. So some of the microbes that you might take as probiotics may be kicked out because you have something that is already occupying that niche. But that can also be advantageous. This happens like with E. coli and Salmonella. Sometimes if you have E. coli present, Salmonella is similar enough that might be occupying the same niche. And so you can actually get into these competitions in a way that is positive. But generally, I think that the point is that we need to learn a lot more about how these ecological systems function and take them as such. You know, fecal microbial transplant is like this very big hammer, and you can be lucky or you could not be. And a lot of times with these more complex diseases, we're seeing that the microbiota just doesn't embed. And I think from an ecological perspective, it makes a lot of sense, right? That you can't just take a community, put it into a new environment and expect for it to be the same community.
So just really quickly, my second question is just a follow-up on that. This science paper from December that focused, or it might have been November, the fecal microbiota transplant in the PD-1 clinical trial, where they essentially do the fecal microbiome transplant and then see an actual response in previously resistant PD-1 melanomas. Do you think that is, I mean, do you have any hypotheses? Is that about the kind of relationship with that mucosal layer? And that when you do that retransplant, now you've got a more, I guess, stable mucosal layer, or do you have any idea about?
Yeah, I thought that was a beautiful study, and you know, it really showed the power that the microbiota holds, and also how, in some sense, like with a microbiota, like it's become in a lot of people this highly traumatized ecosystem. And so, like, for example, in the case of some of these transplants, they were able to work really effectively because the system allowed them to actually re-embed. And when that happens, that can, you know, make the difference between a cancer going into remission as opposed to not responding to therapy, right? But I do think that we need to be careful about how we proceed. It's not, I don't think that the FMTs are going to be a cure-all. We see, for example, for inflammatory diseases that it doesn't seem to be enough. There needs to be either a repeat transplant, so that you're really, again, like with this ecosystem, like you really make sure that things embed. Or they will need to understand more about who's there. You know, when a microbiota is good for me, may not be good for you. The microbiota that I have grown up with has been through all of my history, and it's adapted to my diet. So with just like with things like antibiotics, we often act for a very short-term goal, right? Like we're trying to fix that problem, but we don't know how it's going to affect our health over the long run. I think antibiotics are extremely important because you do want to protect yourself against these pathogens, but you kind of need to, we don't know, like even in the case of those fecal microbiota transplants, what are the long-term impacts that might happen to the host? Obviously, when it's in the case of life or death, like or a cancer, I will gladly get a fecal microbiota transplant if that means that I can cure my cancer. But this is going to be a new microbiota that will have to deal with the immune system and will dictate your health for years to come. So I think it's important to appreciate also that long-term impact on our health in both directions, in the positive and the less positive.
Thanks.
Okay, yep, we have another question from Sarah Dada. If she wants to unmute herself, go ahead.
Hi, Dr. Tropini. It was a good presentation, and it's cool to see what Sarah and Deanna have been up to. But I guess what I was wondering was, I mean, as you kind of said, there's these antigen presenting cells, they work with ILCs, they make these Tregs kind of function, maybe through soluble factors. But if this population, this Mirabacteria, I think it was called, is either being reduced in the number of people it's in and being reduced over time in an ancestry, I mean, do you feel like, I guess I was surprised to hear that it changed the innate, like the immunological response, when it seems like a lot of people have evolved to not use them? Or do you feel like because their genetic profile is different, then they kind of give something that maybe other bacteria don't?
So when you're talking about evolution, are you thinking about the human evolution without them, or are you thinking about the bacterial?
I'm just wondering if there's people kind of trucking along and they have a regulatory response, and it's kind of suggested to reintroduce these Mirabacteria. Like you said, you don't know the long-term response. Like, I guess I'm wondering if the immune response will rebound if this population is gone, or if you know the soluble factors.
That's an excellent question. Actually, exactly what Sarah and Deanna are right now working on. They have this mouse experiment in which we're looking at what happens during the recolonization if you've never been exposed to S24/7, Mirabacteriaceae, or if you have seen it before. And we expect there to be a difference because as you're saying, with the Treg response, it may behave very differently if you have grown with an organism, as opposed to if it just hits you as you're an adult. So stay tuned. We'll hopefully have we'll have some of the data in the next few weeks.
Thanks.
Thank you, Sarah. So here's another question from Kevin Ray. If he wants to say it himself, feel free to interrupt me. But so in the effect of Mirabacteriaceae on the host immune system, is it known whether it's done via direct contact with immune cells, or is it through soluble factors?
Great question. We do not know directly, but we do know that some of the Mirabacteriaceae species that we've been able to isolate go really deep into the mucus. So we do think that there is occasions for direct contact with the epithelium for some of them.
Thank you. And one last question from Amos Rafenia. I believe Amos is left, but are there any other taxa that behave like the Mirabacteriaceae? Sorry, I'm forgetting how to pronounce
No
it.
worries. Yeah, because
yeah.
And also it used to be called S24/7, which makes it a lot easier. So I apologize if throughout the talk I said S24/7 because it probably came out of nowhere for some people. But it's now being called the Mirabacteriaceae, so that's kind of like the proper name that it's going by, and it sure is a mouthful. In terms of other species that are like that, yes, absolutely. We do see that there are other species that become undetectable during following osmotic perturbation, but this was the family that had the strongest response. So in some of the cases, if bacteria start out at low percentages, sometimes it's hard for us to know whether they really went away permanently or whether they're at a low enough level that we're just not detecting them. So we definitely plan to expand our view to also other bacteria that may be less abundant.
Thank you. And one last question we have here from Brahim Sharif. So is it technically possible to count different microbiota from metabolites, and how would that compare to microgenomics?
So by counting the microbiota, like, do you mean actually like one of the ways in which we can count them is by culturing them, so that we dilute them enough so we get to single cells, and then we can count how many there were present in a system. If we want to understand the metabolites, we can do this by doing metabolomics. So we were actually using mass spectrometry to analyze the type of molecules that are present in the gut or also present in the host, so that anything that our microbiota makes it into our blood system because it gets absorbed. And so we think that the metabolites that are present in the serum are very, very important as well. And we're, in terms of whether Microbiology makes things that are important for us, we're very interested in this and something we're actively pursuing. And I'm not exactly sure if I understood the question correctly in terms of the first counting part.
I think if I can try to rephrase, is it possible to identify different taxa through just metabolites alone, or is the use of metagenetic sequencing?
That's metabolites. That's, unless they're very specific types of metabolites, that can be hard. And that kind of gets to the question of what's more important. Is it the genes or is it the organisms? And I think that for the microbiota, is both are true. From the perspective of thinking about the metagenome or the metabolome, of course, from the perspective of the host and what is making it to the blood, the bacteria themselves, or the taxa themselves, are not necessarily as important. But then from the perspective of the immune system, individual species can be really important. In terms of actually being able to identify them, that's pretty hard, because each one of the bacteria that we have in the gut produce hundreds of different metabolites. So the spectra can also change depending on the environment that they're exposed to and the interactions that they have. So that's quite hard to do. That said, there are ways in which you can identify species based on the compounds that are present onto the cell wall. But it's a type of spectroscopy that allows you to identify species based on what is present on their envelope, which is, I don't think, is what you were getting at with the metabolite question.
Thank you so much, Carolina, for joining us today. So it seems as though we've run out of questions and we're running up on to 12:30. I'd just like to thank you on behalf of our whole audience here and on behalf of BandBug for coming and speaking. And for those of you still here, we are still accepting speaker nominations for next season. So feel free to visit vanbug.org to do that. Yes.
Well, thanks so much for having me. Yeah, and feel free to reach out if you have any other questions. My email is on our website, so. Thanks, everyone. Thanks, Will.
Yeah, no problem. Thank you.
Take care.